Now we will be talking about termination in prokaryotes. So this is termination in prokaryotes. Now listen, termination in prokaryotes actually have two well-defined mechanism. Number one, Rho-dependent. Number two, Rho-independent. Clear? Rho-dependent and Rho-independent. Clear? So in prokaryotes, transcription occurs with the help of Rho proteins, or it may be Rho-independent as well. That means no Rho protein is required. Clear? So there are Rho-dependent mechanism and Rho-independent mechanisms. Clear? So there is Rho-dependent mechanism and Rho-independent mechanisms. Clear? Now listen with all your attention. A very important point here we need to understand is this, that in case of termination, we will first talk about Rho-dependent mechanism. Okay? Then we will talk about Rho-independent mechanism. Clear? Okay, so let's start with Rho-dependent mechanism. Okay? So let's start with Rho-dependent mechanism. Now listen. In case of Rho-dependent mechanism, you know that Rho protein will play a very important role, right? Now listen with all your attention. Again, we will have to draw our, polymerase. Now listen, we are always drawing like this, but in this drawing or in this diagram, alpha subunits are, absent. Okay? So this is actually, beta sheet in details. Sorry, not beta sheet. Beta and beta prime pincers are detailed here. So this is suppose your, clear? Now listen with all your attention possible. So we will be talking about Rho-dependent termination, right? Now listen with all your attention. Suppose this is, your so this is 3' of DNA, this is 5' of DNA. So therefore, what is this? This is 5' of RNA and this is 3' of RNA, right? Now listen. In the 5' side, now you listen with all your attention. In the 5' side, there is an important point you need to understand. If you want to, end transcription with the help of Rho protein, you need to have this site. Now listen. There is a special site in, 5' end of RNA, which is coming out of the RNA polymerase. Okay, this site is called RUT site. This site is called RUT site. This site is called RUT site, or Rho utilization site. Okay, this site is called RUT site. Okay, or Rho utilization, clear, or Rho utilization site or RUS site. Okay, now listen. Actually, what happens is this: Rho protein. This Rho protein is actually a hexamer. Okay, and it looks like a donut. Okay, 1, 2, 3, 4, 5, 6. Okay, clear? So this is an hexameric protein. Now listen with all your attention. What is this Rho protein? What is the chemical nature of this Rho protein? Rho protein is nothing but an helicase. Okay, it's an helicase. Okay. Rho protein, this is Rho protein, and this is nothing but a helicase. Clear? Now what happens is this Rho protein actually binds with this right side. Sorry, not right side. This Rho protein, 1, 2, 3, 4, 5. This Rho protein now binds with the RUT site. Okay. Now after binding with the RUT site, what happens is this helicase utilizes ATP. Okay. This Rho protein, these are the characteristics. Okay. They utilizes ATP. Okay. They utilizes ATP. Now what this particular Rho protein is doing is, this Rho protein is actually utilizing ATP is actually utilizing ATP to move towards the this RNA. Now what happens is this, like TRCF protein, I have already told you, like TRCF protein, this Rho protein actually, what it does is, this Rho protein actually gives a blow to the RNA polymerase. It actually literally gives a blow to the RNA polymerase. Clear? It gives a blow to the RNA polymerase, and therefore RNA DNA, this transition, or RNA DNA, this bonding actually breaks off, and the RNA which has been transcribed falls off. Clear? Now, so when this Rho helicase actually now interacts with RNA polymerase, it gives a blow. Now another mechanism is this, that with the help of ATP, with the help of energy derived from ATP, Rho actually moves into this site where there is RNA DNA hybrid. This is the site where there is RNA DNA hybrid. Now this is what? This is what? This is RNA DNA hybrid, right? This is RNA DNA hybrid, right? Now a very important point here we need to understand is this, that this RNA DNA hybrid actually gets dismantled or is disrupted when Rho helicase actually arrives here, and therefore this RNA DNA hybrid breaks off and RNA comes out of this RNA exit channel, and termination of transcription takes place. Clear? Okay. Now listen with all your attention. What about this Rus site? Now Rut site, sorry, why I am telling Rus site? Now, this Rut site is rich in CA, that is cytosine and adenine. Clear? Is rich in CA. Clear? Okay. So this is Rho-dependent mechanism. When you are talking about Rho-independent termination, you need to understand that for Rho-independent termination there are two prerequisites. Okay. Now listen. Those prerequisites are already present in the DNA structure itself. To understand Rho-independent mechanism of RNA transcription, you must understand this very basic thing, that there are two prerequisites. Okay, I have already said that. Now listen. These prerequisites are present on the template strand, but they are also present in the non-template strand. Why? Listen. So, what are the prerequisites? What are the prerequisites of Rho-independent mechanism? What are the prerequisites of Rho-independent mechanism? Now listen with all your attention. The prerequisite number one is very, very important. That there should be alternative base pairing having a gap. Okay, there must be alternative base pairing. Alternative base pairing should be there. Okay, alternative base pairing. Clear? Alternative base pairing. And the second one is the presence of repetition of A rich site in the upstream of DNA. The second point is repetition upstream to the alternative base pairing. Now listen, what is this? Suppose there is TC triple G CG, and then it is followed by N. N means any nucleotide. Okay, N means any nucleotide, and G. Thereafter CG triple CG. Now listen with all your attention. A very important point to understand. And in the upstream, okay, in the downstream, there are A. Okay. Now listen with all your attention. Whenever RNA polymerization will take place, now listen with all your attention. Whenever RNA polymerization will take place, Now what will happen is, whenever RNA polymerization takes place, what will happen is there is what? C, G, 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 C, G, N, right? And then again it is C, G, 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 C, A. Okay. Now listen with all your attention. Now if I can draw the transcript here, you will understand that suppose this is your transcript. Okay. So we have got C, G, 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 G. There are 3 G's and C and G. Then after there are nucleotides again. Then there is C, G, 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 C. Now listen with all your attention. Can you find the peculiarity here? Now listen. What is the peculiarity? This C can now interact with this G. This G can now interact with this C. Clear? And sorry, I have put here okay, I have put here G's. So this will be C. Opposite to G there will be C. Opposite to G there will be C. Opposite to C there will be G. Okay, so there will be 3 C's here. Sorry. Clear? Now listen. So can you find the peculiarity here? Now listen. What will happen is this. Now this whole thing will be converted into a thing like this loop like structure. You know why this loop is occurring? You need to understand this. You know why this loop is occurring? Now listen. This loop is occurring because, now listen, suppose this is your mRNA. Okay, now listen, suppose this is your mRNA. Now there is what? G, then C, right? Then G, 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 C, right? And opposite to C there will be G, C, 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 G, C, right? You will get these things here and here. That means these two are complementary to each other. Therefore, they will have what? They will have hydrogen bonds. Now, important point you need to understand here is this. This loop consists of all n nucleotides. Okay, that means any nucleotides. Clear? But when this stem loop-like structure, or hairpin loop-like structure takes place, now important point you need to understand is this, that when this thing actually comes out, and forms a hairpin-like structure, and forms hairpin-like structure, then you must understand this very basic thing. That means this whole thing. That means what is happening here is this whole these A things have actually now come here, right? These A's have come here, right? Now when these A's are coming here, that means you have a nasty mind, you are laughing now. That means you, right? Now listen. Due to this weight, now listen, this is very important. Okay, this is very important for Rho-independent termination. Why? This continuous repetition of A nucleotides or adenine nucleotides are very important, because these adenine, when interacts with you, they will have what? Double bond, and double bond is weaker than triple bond. Therefore, when this stem loop or hairpin loop like structure will occur, then due to the weightage of these loops, these hydrogen bonds, these two hydrogen bonds cannot hold this heavy thing, and therefore it will fall off. Therefore, this whole thing, this hydrogen bond will break off, and this will all fall off from this RNA exit channel. Clear? And therefore termination occurs. Clear? Termination occurs, and then this RNA polymerase will go out or dismantle, and then DNA will again bound, and they will form again double helix. Clear? So this is Rho independent mechanism. Clear? So in Rho independent mechanism, what we see, that formation of hairpin loop is very important. Clear? So this is the basic thing you need to understand here is this. So these are alternative base pairing. This is what? Repetition of A-rich series or A-rich sites. So these two are very important for Rho independent mechanism. Okay. So we have successfully completed transcription in prokaryotes. Now we will be talking about